Good afternoon, everyone. So my name is Renyu. I'm a Aurora researcher from UMC Utrecht. So we know that exposure assessment is fundamental to risk assessment, and is especially relevant for human research because for toxicology research, for example, using cell lines or animal models, we can easily use particles, for example, made by Lawrence to these cells or models, but this is not possible with the human. So my talk today will be focusing on the complexity of exposure assessment of micro nanoplastic, especially in the context of human research where human biological samples are analyzed to quantify and characterize micro nanoplastics that could reflect internal human exposure. So when we look at the bigger picture, so we know that outdoor environmental samples, for example, the marine systems and soil and air, they were the initial focuses for micro nanoplastic detection. And recently, more attention has been given to indoor samples, including household plastic, like Amanda just introduced in her talk, because we think indoor exposure could be more relevant to human exposure. However, environmental exposure only provides indirect evidence for human exposure, so that emerging studies have also investigated micro nanoplastic in human samples. But we need to also be we will also need to realize that compared to environmental samples, human biological samples involve an extra layer of complexity. For example, human biological samples are more subject to, for example, smaller quantity or volume of samples, storage conditions, for example, including temperature and duration, and also potential biological interference, and also potentially inappropriate digestion procedures, which are always essential for human samples, and also relatively low realistic concentration, and also more limited analytical sensitivity. So in this slide, I show a typical analytical pipeline for micro nanoplastic assessment in human tissues, where we typically start with sample collection from participants. And most of the time, they are not analyzed immediately after collection, so that they also need to be stored properly with a certain container within a good condition. And most of the time, they also need to be shipped to a dedicated laboratory with specific specialized with some expertise in micro nanoplastic assessment, where precautions need to be taken to minimize potential contamination. And because biological samples also compresses a lot of substantial amounts of organic compounds, so that they need to be removed. So in the we call pre-processing steps, we do matrix digestion and also filtration or residue isolation to get rid of this organic compounds as much as possible, and then to also concentrate the plastic particles. After which we can do micro nanoplastic assessment using different methods, which are broadly categorized into two groups, which is microscope-based and mass-based method. So microscope-based method, for example, Raman mass microscopy or LDR or PIR, they can be used to characterize the feature of microplastic particles, for example, morphology like size, shape, pigmentation, or surface properties, but also to identify polymer type and also to give information on the concentration by counting the number of the observed particles per sample unit. On the other hand, mass-based method represented by pyrolysis GCMS, they can be used to also identify polymer type and the concentration, for example, a microgram per sample unit. But because it doesn't rely on observation of the actual particles, so they cannot provide information on the physical property. And despite all the because there could be contamination in all the steps of this workflow. So we also need to think about adequate QA quality assurance or quality control measures, which is QA/QC. For example, we normally need to have a negative control, which are, for example, like procedure blanks analyzed together with the samples so that we know like how many potential contamination we may have until from the final result. And also it's essential to have positive controls where we spec with spike metrics with a known quantity and polymer type of particles to evaluate the recovery rate and also the overall workflow performance. So in the table on the right, I also show, besides contamination, which I have mentioned that can occur at every stage of the workflow, There could also be other potential bias, for example, particle loss during the digestion and filtration phase, for example, because of the pore size of the filter. And also in the micro-nanoplastic assessment field, because especially like if we use microscope-based methods, so different methods, they have their specific resolution. So if the particle size fall below the minimum resolution of the method, then they cannot be really observed. And also there could be analytical interference because of the residue of the digestion chemicals or because of the biological material, especially relevant to mass-based method like pyrolysis GCMS. And also there could be measurement bias, mostly concerning digestion, for example, selected polymer degradation, filtration, for example, clogging issue, and also micro-nanoplastic assessment process, for example, due to spectral mismatching or polymer misidentification. And here I show, I summarize the most commonly used technique in the human empirical study published by the end of 2025 about assessing micro-nanoplastics in human tissues. So we can see a clear increased amount of publication over time, mostly after 2023. And if you cannot see here, I can show you that it's quite obvious that microscopy-based methods represented by the red color remain predominant in the field. However, you can also see an increasing trend of using mass-based methods, for example, pyrolysis GCMS represented by the blue color, as well as research combining both methods in the same context, for example, represented by the color purple in this figure. And there are some challenges and debates about micro-nanoplastic assessment in the human tissue, not only from academia, but also from the general media. For example, the science paper published in 2024, and also the famous Guardian article that came earlier this year, which both have some concern or criticism on the contamination control, as well as some critiques on the pyrolysis GCMS results because of the potential matrix interference, especially for PE and CBC, because they can be interfered by some signal from biological material like a lipid or protein. And because pyrolysis GCMS also not provide information on morphology, so that's also a lack of confirmation of some particle properties. And there's also quite some variabilities of the result from, for example, micro-nanoplastic in blood and urine. The result can differ a lot across different studies, partially because of the lack of standard operation procedures and also inadequate QC recommend practice. And there's also some concern about biological possibility because most of the time, especially using microscopy, the particle observed are actually microplastics. However, if you think biologically, maybe a particle of smaller size, for example, nanoplastic, they have a bigger potential to translocate in the human body. But because of technical limitation, they are less investigated compared to microplastics. And recent effort has been made to propose some QC framework for micro-nanoplastic exposure assessment relevant to human exposure, partially building upon established QC framework for environmental samples, highlighting some key aspects. For example, measurement to minimize contamination, and repeated measurement to enhance reproducibility and precision. And as I also mentioned, negative control and positive controls should be set. And also we need to be transparent about the reagent, including solvent, surfactant, and also digestion chemicals that we use, and also the concentration, and maybe also under which condition they were used. And also the particle size range, also because it can be subject to many factors, including the method we use, but also the digestion and also the filtration process. And also the key parameter used, for example, for pyrolysis, we need to also make note like which reference material we use and which calibration standard we use. Also the LQ, LD information are also crucial. And also, we also need to recommend, it's also recommended to combine microscopy-based method and mass-based method in the same study to complement results of each other. So let's bring the story back to the AURORA to see what we have done to do to achieve better QC of our study. So besides standard laboratory procedures, we also do analyze duplicated samples to make sure that we have representative or reliable results. And also we do a de novo collection where we we follow a protocol where minimize plastic contamination using, for example, containers of non-plastic materials, and also to make sure that everything is contain the least plastic material as possible. And we collect samples from mother and child paired sample from mother and children, for example, placenta tissue, cord blood, and maternal plasma and maternal urine. And we can compare, because historically the sample we collected, there's contamination is inevitable. So we also want to compare the result, whether that it's substantial difference in the result, so that we can infer whether there's a potential part of the result come from contamination or not. And then we also perform special distribution of the placenta, because we know very little about whether a specific part of because we always analyze a small proportion of the sample, but we don't know whether the micronanoplastic concentration in this specific proportion can be representative of all the entire organ, not only for placenta, but almost for every other liquid, for example, blood, urine. We don't know the distribution of the microplastic in this matrix. And also we perform independent lab validation using different methods. For example, primarily we use pyrolysis GCM mass and to analyze micronanoplastic placenta tissues in Emory. And also we took a subset of sample to analyze in the same method in German laboratory. And we also took a subset of sample analyzing using different methods, for example, microscopy-based method, also TR, PCR, and MS method, to see how the results align with each other. And also we made some effort to investigate potential metric effect metric interference. For example, how if we can analyze signal of, for example, lipid and protein, and then we think we can subtract the signal from lipid and protein from the total, so that we know that maybe there can be a way to minimize this interference. And also in the analysis, we may also want to prioritize particles that polymer that we know are maybe less influenced by those interference, so that we know that our results are more reliable. And so to conclude my talk, so it's complicated to do exposure assessment of micronanoplastic in human tissue, and adequate QA/QC consideration and also transparent reporting should be recommended to improve study quality, and also to enhance reproducibility, but also to achieve more robust evidence synthesis. Thank you very much.